good afternoon, everyone. Welcome back. So this is our final session today. Thank you again to Peter, who is busily answering whole dozens and dozens of questions in the chat. I hope that can continue. So I'm going to introduce the last session today. So these are the fellows in the HBBE connected to the living construction theme. Slight reordering of our presentations today because we've got a couple of technical problems in the background. So first I'm going to introduce Yunhong, who brilliantly is sharing his screen already. Feel free to turn on your camera as well, Yunhong, at this point. And well, I'll let you go for it.
Thank you, Martin. Can you hear me?
We can hear you perfectly. That's great.
Good. Hello, everyone. Thank you for coming. My name is Yunhong Jiang. I'm a Vice Chancellor Fellow in HPV Group, and welcome to this HPV Avenue Summit 2021. Today, I would like to talk about the fabrication of the functional bio-based materials for thermal insulation and the building environment. Firstly, I would like to give a brief introduction on my research. My research is mainly focused on design and synthesis of nanomaterials for different applications, including antimicrobial surface, hydrophilic and hydrophobic surface, and the bio interaction between bacterial cells and the nanomaterials. I also have a very extensive experience on application of the bio-based materials for building insulations from macro scale to bulk scales, and the application of the porous materials as a solvent due to high surface area and tunable structures. Follows the Peter speak, we can see use the biology synthesis, we can still produce a nanoparticle as well, shown in this slide. And you can use the bacterial cells. You can use the plant cell extraction. So I will talk more later. And in the HPV group, I'm working in living construction group. As you have heard from Meng and Martin this morning, the objective of living construction group is to develop a new generation of the engineered living materials through biology process. We mainly work on biominerals, biopolymers, and the responsive materials, which can be used in structural components, insulations, and barrier memories. In the group, I'm mainly work on functionalization of the biomaterials for different building applications. Here I'm going to talk about some work on biopolymers of the bacterial cellulose. Why we try to use the bio-based material in the building? As we all know that currently there is an interesting attention on energy efficient and cutting carbon emissions from our homes due to the concern about the climate change. Heating and cooling in the building account for over 10% of global energy consumption. In addition, the manufacturing of the synthetic insulation materials is an energy-intensive industries. There are possibilities for bioengineers to generate or develop the super insulation product using bio-based materials with good ability for moisture and vapor management to improve thermal efficiency and building comfort. So in next few slides, I would like to talk about one project we are working on to develop the functional bacterial cellulose aerogel membrane with a lower thermal conductivity and some functional surface for building insulations. As we all know that the bacterial cellulose can be obtained as a membrane by a single growth of bacteria. The growth mechanism of the bacterial cellulose has been well studied, showing top pictures. Some of research already showed that the bacterial cellulose aerogel and hydrogel can be achieved by a single step of the solvent exchange. Membrane morphologies depend on the shape of this interface, which can be manipulated or controlled during the synthesis. The bacterial cellulose product have strong flexibility and show high tensile strength and a high degree of the polymerizations compared to plant cellulose. They are high chemical stability and have very high adsorption capacities due to their high porosity and large surface area. All these special properties make them have very widely applications. They have been widely used in medical application and food packing. But despite all of the attractive property of the bacterial cellulose, its product has been not bound to building insulation applications. There are several problems limited their practical applications using in the building environment. Firstly, the bacterial cellulose is highly hydrophilic nature, resulting in very high liquid loading capacities. They are easy to biologically decay if exposed to unsuitable environment conditions. The second issue of using bacterial cellulose as a source of the insulation materials is the problem with fire due to their very low thermal stability. There is a huge potential to develop no functional bacterial cellulose with new features for more diverse applications. Some various modification methods have been employed to achieve additional functionality to give bacterial cellulose new features. Due to time issues, I don't give too much information about chemical modification. Generally, the physical and chemical surface property are crucial factors in the interactions of the material with the surrounding tissues. The morphological modifications normally means increase the roughness, shifting topographies from micro to nanoscales on the surface of the materials. All these modifications can give specific properties like change the hydrophilic surface to hydrophobic, or change the material to antimicrobial surface. And the functionalization means to introduce a certain functional group on its surface or change its own surface chemistry. We can also modify the bacterial cellulose during the growth. We can call the in-situ modification, or after growth, we can call the ex-situ treatment or post-treatment. Currently, my PhD student Chris Milner are working on in-situ modification of the bacterial cellulose using silicon and acetylate. You can find more information from his poster. In the project, we try use the soil method to modify the bacterial cellulose surface. This slide shows the soil hydrolysis polymerization diagram. The soil method has been well studied and widely used in introducing different functional groups to different substrates, as you can see from the graph. The final products can be produced through the coating, gelling, or precipitation or heating approach. It can be in dense films, aerogel, uniform particles, or ceramic fibers. The different functional group also can be added into the process to change the productive properties. So the project we developed is use functional soil gel hydrophobic coatings. The MTS hydrophobic functional group can be added into the tessel solutions. Then the functional alkaline chain can be bound to sugar unit through hydrophilic silica network. The modified functional soil gel can change the surface of the hemshift from hydrophilic to hydrophobic by dipping coating, as shown in the photos. The XPS data show the different bonding energy on the surface before and after modifications. There are also many ways to change the surface antimicrobial surface, such introduce the antimicrobial agent or grafting antibiotics or produce the nanostructure patterns on the surface. So I'm not going to discuss more here. If you like, I can discuss later. In our project, the key technology will be developed is to modify the bacterial cellulose to render them more resistant to biodegradation through in-situ bioprocess approach. The bacterial cellulose will be in-situ modified with improved functionalities, including hydrophobicity, antimicrobial activity, and fair resistance. Several approaches will apply including developed silica bacterial cellulose aerogel or incorporate sodium silicate or co-culture bacteria to produce specific nanoparticle into the bacterial cellulose. Due to the COVID-19, the project are still in the very early stage, so I don't have too many data to show here. Certainly, we also would like to work on scale-up process using food waste to reduce the cost. As we can see from the insulation property of the porous material comes through a combination of the two characteristics. The material's natural capacity is to inhibit the transition of heat and use the pocket of the trapped air. So we will also try to optimize the biological process to enhance the efficiency of the fabrication of the materials and controlling the physical properties and porosity with the tunable porosity to achieve the highest possible thermal insulation values as well. The developed bacterial cellulose insulation membrane will be characterized for their microstructure, densities, porosity, and surface chemistry. The characteristics are more or less linked to hygrothermal and the mechanical property of final product. Their thermal property, mechanical strength, hygraic property, and durability will be studied using multi-scale analysis techniques. The final developed bacterial membrane will have a super low thermal conductivity and resist to biodecay and fire. It can be used for insulation, air purification, fire retardance, and sound insulation. Hopefully, the final product can be a good competitor compared to silica aerogel and the plant cellulose aerogel on the market. So in this slide, I just briefly show a collaboration project. The result has been published in Journal of Carbon Dioxide Utilization 2020. We have successfully synthesized a carbonic anhydrase hybrid ketone hydrogel membrane for carbon dioxide captures. The amount of the calcium carbonate produced by development member was 9 times compared with free carbon anhydrase. The developed membrane show a higher thermal stability, storage stability, and pH stability than free carbonic anhydrase. And this slide, we also use the ZIF MOF material to encapsulate the carbonate anhydrase, FIT-DH, NADH, and GDH for enhanced formate yield rate. The system shows super capacity for conversion of carbon dioxide to formate. The detailed experiment can be found in the paper published in the International Journal of the Biological Macromolecules 2020. So, based on 2 collaboration projects, I'm also interested in developing the bacterial cellulose hydrogel membrane as a nanoporous carrier for improving indoor air quality for carbon dioxide capture. The developed bacterial cellulose membrane incorporates ZIF-8 MOF material or engineered bacteria can be used for filters to achieve high efficiency filtration through electrostatic adsorptions. The tailor-made bacterial cellulose membrane also owns efficient antibacterial potency to keep the human body from infection germs. To summarize, my research mainly focused on using bio and nanotechnology to develop and improve the bio-based materials properties for the thermal insulation, indoor air quality, and antimicrobial solutions in the building environment. Please feel free to contact me through the email below, and I look forward to speaking with you later. Thank you.